Hello, good evening. And good evening, everyone. We are from Group 5, and today we would like to present our PBL2 Biochemistry on the topics of carbohydrates and lipids. My name is Aufar. I'm with my group with Alissa, Rizqi Naufal, and Nikita Kanjani. Next. Be presented by each and every one of us, explaining on the subtopics on this topic.
Okay, so for the first topic, how modern diet increases the prevalence of obesity in Indonesia and Mexico. So obesity is driven by complex interactions between genetic, behavioral, environmental, and socioeconomic factors, with rapid urbanization and globalization accelerating the shift toward high-calorie diet and sedentary lifestyles. Research shows that genetic predisposition plays a role in obesity by influencing mechanisms of appetite regulation and fat storage. However, these genetic factors usually require environmental triggers such as high in fat and sugar diets to fully manifest. In Mexico and Indonesia, cheap and accessible ultra-processed foods and sugary drinks dominate our diets, while healthier options are less affordable, and advertising further promotes their consumption. At the same time, urbanization has reduced physical activity through sedentary jobs, motorized transport, and limited exercise, creating an imbalance between high calorie intake and low energy use that drive obesity in both countries. So do I agree with government implementing sugar tax? Yes, I do, because evidence shows that it can reduce unhealthy consumption and improve public health. In Mexico, a 10% tax on sugar sweetened beverages led to a 6 to 12% drop in purchases within 2 years, especially among low-income groups, as you see from the graph here. And by drinking, chronic related metabolic disorders. This is especially relevant in Indonesia, where obesity prevalence from 10% 2007 to 21.8% in 2009, triggered to help reduce future healthcare costs. And also the World Health Organization support sugar taxes as one of the most effective strategies to combat obesity. And why does the industry strongly oppose a sugar tax? The food and beverage industry profits from addictive sugary products that stimulate dopamine-driven rewards pathways that encourage repeat consumption. For this reason, they oppose sugar taxes mainly out of economic interest, fearing reduced sales and profits. They also argue that such taxes could cause job losses in the sugar, beverage, and retail sectors, while using marketing power to frame sugar taxes as unfair, overprotective, and burdensome for consumers.
So, we will discuss on the question of sugar-free products. Replace sugar with artificial sweeteners. How does the body process these artificial sweeteners compared to natural sugar? And why is the metabolism of artificial sweetener different from that of regular glucose? Can consuming large amounts of artificial sweeteners. So the subtopics that we have three, which is what is artificial sweeteners and natural sugars, the metabolism of artificial sweeteners and natural sugars, and metabolic disorders caused by artificial sweeteners. So first up, what is natural sugars? Natural sugars are organic compounds that occur naturally in foods, classified as simple carbohydrates, generally monosaccharides and disaccharides. Those are the two types of natural sugars that come across daily. It acts as the primary metabolic substrates for consumption in. So is disaccharides. Disaccharides is something formed carbohydrate formed by the linkage of two monosaccharides through glycosidic bonds. And what is monosaccharides? Monosaccharides is the simplest form of saccharides. Uh, and these are the examples of disaccharides, which is sucrose and lactose. Then the example of monosaccharides are D-glucose, D-fructose, and D-galactose. and these are the structures of each forms. Next up is artificial sweeteners. What are they? They are not sugars, but they are actually chemically substances which are produced in labs and they do not occur naturally. So there are these types of artificial sugars, which is saccharin, aspartame, acesulfame K or ASK, and then you have neotame and sucralose, and as well as HS. And these are the forms of artificial sweeteners. Next up is how the body processes artificial sweeteners and natural sugars. So uh, these are the processes that occur where in natural sugars, first of all, you have taste detection, and then second, you have the digestion, third, you have the absorption, fourth, you have the metabolism, fifth, you have the blood sugar regulation, sixth, you have the energy use and storage, and seventh, you have the minimal excretion. As for artificial sweeteners, it is shorter. As from first of all, you have taste detection. Then second of all, you have the digestion and absorption. And then you have the metabolism. Then fourth, you have the insulin and hormone effects. And fifth, you have the excretion. So what actually happens between natural sugars and artificial sugars when they are processed in the body is that natural sugars are actually used for energy with direct effects on the blood glucose, whereas the artificial sweeteners provide taste but no calories, so they undergo minimal absorption, where they provide little to no energy. Next, we'll be covering the metabolism of artificial sweeteners and sugars, where natural sugars and artificial sweeteners have a difference where in energy production, as mentioned before, that natural sugars produce energy, whereas sweeteners produce little, and then the absorption is by the cells, whereas the artificial sugars sweeteners are uh, not absorbed. Then you have the insulin and blood sugars, where for natural sugars raises glucose, stimulants, and insulin, whereas for artificial sweeteners, there's minimal effects on blood glucose and insulin. Then in the gut interaction, there's normal glucose absorption and gut response for natural sugars, and for artificial sweeteners, there's gut receptors or microbiota. Sugars is for major energy source, whereas the artificial sweetener is mostly inert. The blood by artificial sweeteners, three main things that occur, which is the biosis and physiological repercussions, as well as health implications. Where focusing mainly on the health implications, where there's great risk to obesity, and then there's also increased likelihood of metabolic syndrome, and higher interest on higher risk to have diabetes. So the key takeaways are that artificial sweeteners can help reduce sugar or calories, but have a use especially in ultra processed products, which may contribute to metabolic disorders.
Okay, so the third question is carbohydrate plays a central role in energy synthesis in the body. Explain the process of carbohydrate metabolism from ingestion in the oral cavity to metabolism in cells or cellular respiration. So there are five key points that will be covered in this one specific question. Next. Okay, what is carbohydrates? Almost similar to what Aufar has mentioned before, carbohydrate generally consists of cis-skeletal monomer bearing C double bond O, OH, and sometimes NH2. It is the fuel that keeps the brain and nervous system functioning. So there are three different classes of carbohydrates. First is sugar or simple carbohydrates. Secondly, starches or complex carbohydrates, and finally fiber, which is indigestible carbohydrates. And here I include some example for each, like fructose, sucrose for simple, cellulose, cellobiose, and amylose for complex, and vegetables and fruits and seeds for fibers. Okay, next. Okay. So how does carbohydrate get metabolized? Firstly, it gets broken down into monosaccharides or sugar or carbohydrate with one sugar chain. So firstly, the mouth secretes amylase enzyme to break down carbohydrates into glucose, and then it will continue until it reaches the stomach, where the stomach acid stops the uh, enzymatic reaction, and uh, then after it passes through small intestine, the pancreas secreted amylase into small intestine, which break it further to be finally readily absorbed into the bloodstream as glucose and used for cellular respiration. Next. Okay, so there are three different step in cellular respiration. Firstly is glycolysis. Glycolysis is the hydration, so hydrolysis process where glucose is broken down into two pyruvate acid, which firstly the glucose gets phosphorized using 2 ATP, and then finally at the end uh, uh, 2 pyruvate acid is generated, and they will get used in Krebs cycle. In Krebs cycle, there are 8 steps, and uh, in this Krebs cycle, the byproduct is water and CO2. And finally, is the oxidative phosphorylation. This is where the ATP gets produced. The NADH and FADH produced in Krebs cycle is generated is used to generate the ATP, and the product of this step is 38 ATP produced, or adenosine triphosphate. They will be used in for like the currency in our cell to be functioning well. And yeah, next please.
Okay, so right now we're going to be talking about the absorption in our body. So, as mentioned earlier by Naufal and Aufar, simple carbohydrates are composed of one or two sugar units known as monosaccharides like glucose, fructose, and galactose, and disaccharides are sucrose or lactose. And because they are structurally simple, they require little to no digestion before absorption. And in the small intestines, enzymes such as maltase, sucrase, and lactase rapidly break down the disaccharides into the monosaccharides. And these monosaccharides are then absorbed into the intestinal cells and transported into the bloodstream for usage. Once glucose enters the blood, it travels to tissues and cells where it serves as the main fuel for energy through processes like glycolysis, the citric acid cycle, where it then undergoes cellular respiration. Glucose is then converted to ATP, which is the energy molecule that powers nearly every single cellular activity in our body. And because this process I just mentioned happens very quickly in our body, simple carbohydrates provide a rapid energy boost. However, this energy spike doesn't last very long because after the glucose surge, blood sugar often drops below line, leading to hunger shortly after eating. Next slide, please. And since blood glucose can rise and fall so quickly, Basically, the body maintains homeostasis through the secretion of two hormones, glucagon and insulin. The key organ responsible here is the pancreas, specifically the islets of Langerhans, which contains two types of hormone-secreting cells. The alpha cells, which secrete glucagon when blood glucose falls, and beta cells, which secrete insulin when blood glucose rises. And these two hormones work as a feedback system to keep glucose levels stable. Next, please. So this slide explains how when glucose levels increase after a meal, beta cells insulin, signaling cells to absorb glucose. Conversely, when glucose levels drop during fasting, exercising, etc., alpha cells are going to release glucagon, which stimulates to convert the stored glycogen into glucose. Next, please. And this is just a visual diagram of the process I explained earlier, which is the feedback loop of how our pancreas rotate between secreting glucagon and insulin. Next, please. Next aims to answer how a high carbohydrate but low fat can cause So during exercise, our muscles mainly use the glycogen, which is the stored form of glucose, as their primary energy source. However, glycogen reserves are very limited. So within 60 to 90 minutes of continuous movement, our glycogen stores are depleted completely. And once these glycogen stores are completely exhausted, the body must rely on fats for fuel. But if someone follows a low-fat diet, their ability to oxidize fat through the beta oxidation process becomes impaired because the metabolic enzymes that are responsible for the fat breakdown becomes much less active. As a result, the body cannot efficiently switch from carbohydrate to fat metabolism, leading to early fatigue. And this condition is known as limited metabolic flexibility because they have difficulty in converting, in changing from using carbohydrates as a fuel to fats. Next, please. So this slide focuses on how the metabolic flexibility in our body of how we can switch between using carbohydrates and fats as fuels. So during the fed state, as in right after eating, insulin levels are really high, and glucose is readily available, and the body rises the glucose present in the bloodstream for ATP production through glycolysis and oxidation. And the excess glucose is stored as glycogen or converted into fatty acids and stored as glycerol. And at this stage, the lipid metabolism is suppressed because high insulin levels inhibit the breakdown of triglycerides in the adipose tissue. However, during fasting or next, please. During fasting or energy demand activities, insulin levels falls and glucagon rises. The body then shifts using fats as its primary energy source, breaking triglycerides down into free fatty acids, and glycerol. And in this slide, there are also details that explain what happens in the early and later phases of fasting. To summarize, in the early fasting phase, which is 12 to 24 hours after the last meal, the liver still maintains the blood glucose through glycogenolysis, which is breaking down the glycogen into glucose. However, during prolonged fasting, which is beyond 24 hours, the metabolism shifts even further. The liver then increase lipolysis as soon as the glycogen stores are depleted, breaking down the triglycerides into free fatty acids and glycerol. These free fatty acids then enter our bloodstream undergo yes. Body chooses energy sources during different carbohydrates because they're the ones that break down the quickest. And this activity usually leads to the formation of lactic acid because our body and cells undergo anaerobic respiration. And at moderate intensity, energy comes from both the carbohydrates and fats because the oxygen availability is sufficient for our cells to undergo or for aerobic respiration to dominate. And during low intensity and long duration activities like walking or cycling, the body primarily shifts to fat metabolism because our lipids are oxidized through beta oxidation to generate ATP more slowly but for a longer duration. And this process conserves the glycogen for essential organs such as brain and red blood cells. So to summarize, the type of fuel the body uses depends on the intensity of the exercise, duration, and oxygen availability. Carbohydrates provide rapid energy, which is why it's used in high intensity, short duration exercises, while fats ensure and long ability.
Okay. And the sixth question is, what is enzymes? Enzymes is a biological catalyst that helps speed up the breaking down process of many nutrients, including carbohydrates. Amylase is the enzyme that breaks down carbohydrate into glucose. And amylase is secreted by the salivary gland in the mouth and pancreas in small intestine. Its primary role is to break glycosidic bond, transforming complex carbohydrate into simple sugar. Next.
Okay, so for the last question, what is the differences in the metabolism of saturated fat, unsaturated fat, and trans fat? So the metabolism of fats involves breaking down triglycerides into glycerol and fatty acids through lipolysis, then converting acids into acetyl-CoA via beta oxidation for energy production in the Krebs cycle. So the differences between these fats are on their structure, which for saturated fat is the structure, they have a three-chain structure. Clicking double bond is the room temperature. They can raises your LDL cholesterol, increases lowering your and increases your atherosclerosis and cardiovascular risk. And then next for unsaturated fat, they have one or more double bonds in their hydrocarbon chain, causing them to be bent and liquid at room temperature. This structure leads to looser packing of molecules because of the double bonds. Unsaturated fats yield slightly less ATP than saturated fats of the same chain length. Unsaturated fat have two types, which is monounsaturated. They only have one double bond. And for polyunsaturated, they have at least 2 double bonds in their hydrocarbon chains. For unsaturated fat, they may lower your LDL cholesterol or increase cholesterol and risk of heart disease. And the last one is trans fats. It is an unsaturated fatty acid with at least 1 double bond in the trans configuration, where the hydrogen atoms on the either side of the double bond are on opposite sides of the carbon chain, creating a straighter molecular shape and makes it more similar in physical properties to saturated fats, making them solid at room temperature. It also can raises your LDL cholesterol, lowering your HDL cholesterol and clog arteries, and increases the risk of type 2 diabetes and heart disease and stroke. So while some fats are considered healthier, the presence and type of double bond in fatty acids determine whether the chain is bent or straight, which directly affects their properties and health impact. Like for the cheese unsaturated fats, it contains of double bonds that create bends in the chain, preventing tight packing, which results in more fluid membranes and healthier metabolic effects. While for saturated and trans fat, they have straight chains that pack tightly together, making the membranes more rigid, and this rigidity alters the metabolism. So these are our references. Thank you.